Hi everyone, and welcome to our last screencast about transport across the membrane. Let's start by asking ourselves what we know so far. We've talked about diffusion, passive diffusion, where molecules can just go across the phospholipid membrane, or they can use a carrier going from a high to a low concentration. Let's break that down a little bit more. So simple diffusion, we know, is when molecules move from a high concentration across the membrane towards a low concentration. So anything that's small, that's not polar and not charged can move this way. So we talked about oxygen, CO2, fatty acids, glycerol, alcohol, urea, and it doesn't take any energy. So if we look at this phospholipid bilayer membrane, we draw a little oxygen molecule. It's small, it's not charged, it's not polar, and it can go straight across that phospholipid membrane. Of course, it won't take energy if it's moving from a high concentration towards a low concentration, where there's a lot of it, towards where there's not very much of it. And when we say that we're moving from a high concentration to a low concentration, we can also say that the molecule is moving down the concentration gradient. And all that means is it's going from high to low. It's moving down the concentration gradient. So how can we make diffusion happen faster than it does naturally? Well, the first thing we can do is we can increase the temperature, because we know that molecules move faster, they collide into each other when the temperature is higher. The second thing we can do is we can increase the concentration gradient. We can put more molecules where there's a high concentration, so diffusion will happen faster. And lastly, we can digest bigger molecules into smaller molecules, because of course if you're small you'll be able to move more quickly across the membrane. The second thing we learned in our last screencast was about osmosis. Osmosis is the diffusion of water. It requires an ion channel because of course water is polar, so it can't go across the fat layer. And of course water moves from where there's a high concentration of water to where there's a low concentration of water. And there's a low concentration of water wherever it's salty. So water always moves towards the salty solution, or you could say that as towards the area with more solutes, or towards the hypertonic area. This does not take any energy, and we talked about three different types of solutions last screencast. We talked about the hypertonic solution. So hypertonic solution is a really salty solution, and if you look, I'm drawing here, the salt is outside of the cell, it's in the salty solution. So the solution surrounding the cell has a lot of stuff in it, a lot of solutes, it's hypertonic. And so the water is going to leave the cell and move out towards that salty solution, and therefore the cell will shrink, get smaller. And we learned also that when a cell shrinks, that's called crenation. The second type of solution that you learned about is the hypotonic solution. Now, hypotonic solution is a dilute solution. So here you've got a cell sitting in a watery solution, so the salts inside the cell are more than the salts or the solutes outside the cell. So there's more stuff in the cell, so water from the dilute solution moves into the cell to dilute it. And the cell gets bigger and sometimes will burst, and we learned last day that that's called lysis. The last type of solution is the isotonic solution, where the amount of salt or solutes is the same inside as it is outside. They're equal concentrations. So the water's still going to move. It'll move in and out, but it'll move in and out equally. So the size of the cell stays the same. It doesn't grow, it doesn't shrink. And in all these cases, the water will move until it's equal, or at equilibrium. So let's talk about facilitated diffusion. Facilitated diffusion basically means that you're going to need a protein to help you move in and out of the cell. There's two types of proteins that we learned about. The first type is the protein ion channel. The second type is the protein carrier molecule. The ion channel is like a tunnel, and the protein carrier molecule changes shape as it moves different molecules in and out of the cell. And of course, the things that have to move this way are things that are larger, things that are charged, or things that are polar. And these are molecules that will need facilitation. And if you're moving from a high concentration to a low concentration, then we call that passive transport. Facilitated passive transport. Moving from high to low, but you just need a little help. You need some facilitation. So what's actually going to move this way? What is polar, charged, or too large? Well, a polar molecule is a molecule like water. The charged molecules that would move that way would be charged ions like sodium, potassium, calcium, magnesium, etc. And the molecules that are too large are some of the monomers that we've talked about, like here's a simple sugar, a monosaccharide like glucose, ribose, deoxyribose. That would move this way and if facilitated with a protein. If you took that sugar and you added a phosphate group and an A, C, T, or G nitrogen base, then you'd have the monomer of a nucleotide. So nucleotides move this way as well. And lastly, if you had a carbon attached to an amino group and a carboxyl group, a hydrogen and an R group, then you've got an amino acid. And amino acids are another monomer that move this way. So sugars, amino acids, nucleotides are all the monomers that move with the help of a protein, and of course ions and water as well. And this is still the same as diffusion, so you're moving from a high concentration towards a low concentration. And you need the help of a protein carrier or a protein channel. There's no energy required because you're moving from high to low, down the concentration gradient. So you see here a protein ion channel like a tunnel, and up the top left you see the protein carrier. Let's take a look at one type of a protein ion channel. This is in the nerve cells. So you see once the gates open, the sodium will flow from a high concentration to a low concentration into the nerve cell. So there was a high concentration of sodium outside and a low inside, so it moved into the cell. And that does not take any energy. So where is facilitated transport important? Well, just like I showed you, it's important in nerve cells. It's also important in the intestines when you're absorbing all those monomers from your food. Sugars, amino acids, nucleotides. It's important in your muscles. Muscles need calcium. And it's important where the muscle cells meet the nerve cells. So let's talk about active transport. Active transport does need energy. And the reason it needs energy is because you're forcing things against nature. You're forcing things to move from a low concentration towards a high concentration, shoving it away that it doesn't want to go, against the concentration gradient. Or that's sometimes called uphill. You're trying to move molecules uphill. It takes a lot of energy to do this because they don't want to go that way. And so it requires energy in the form of ATP. So here we see with diffusion, you're moving from high to low across the phospholipids. No energy needed. With facilitated diffusion, you go from high to low. You need a protein helper to do that. No energy is needed. But if you're trying to go from a low concentration to a high concentration, usually with a channel or a carrier, then you do need ATP energy, and that's called active transport. So here's an example of active transport. If we just watch, these red molecules are being shoved inside the cell with the help of a protein carrier, even though there's already tons of red molecules inside the cell. We've got a low concentration outside, high concentration inside, but you're still shoving things towards the inside. Here's a clear picture of the sodium-potassium pump. This protein carrier actually forcing the sodium to go outside, then the potassium uses the same carrier to be brought inside, and both of those are being moved against the concentration gradient. This happens in the nervous system, and we'll talk more about that when we get to the nervous system. So where is active transport important? Again, the nervous system. Also, though, a really good example of that is the thyroid gland, because the thyroid gland requires a lot of iodine. So even though there's tons of iodine in the thyroid gland already, and not very much in the blood, we're still constantly shoving as much iodine as we can into the thyroid gland. From a low concentration in the blood to a high concentration inside the thyroid gland. And that helps with our metabolism. The last thing we'll talk about today also requires a lot of energy, and that's endocytosis and exocytosis. And the reason we need a lot of energy is because we're moving some really big molecules, much bigger than any we've talked about so far. So we need energy to do this. So we get that ATP from the mitochondria. Endocytosis, you can think, means in. Exo means out, just like an exit sign. So if you're taking a vesicle and dumping the contents outside the cell, it's exocytosis. Bringing contents in the cell, that's endocytosis. There's two types of endocytosis. phagocytosis, which we also call cell eating, where you're moving really big stuff inside a cell. For example, a white blood cell phagocytizing or eating a bacteria. That's a really big item to be moving inside you. And pinocytosis happens on a much smaller scale. We call it cell drinking. And this happens in the intestines. We also call it intestinal sipping. So here's phagocytosis. You see your white blood cell, or this is an amoeba, which is very like a white blood cell. And there's the bacteria. And the white blood cell is going to bring that bacteria into its body by forming a vesicle around it. So here you see the amoeba, and it's going to eat by bringing the vesicle inside its body. That's phagocytosis. Pinocytosis is on a much smaller scale. It's not eating an entire bacteria, but it's bringing smaller stuff inside a cell. So the stuff is smaller, but it's still too big to be moved across just with a protein carrier or a protein channel. So it's going to need a vesicle to bring it inside the cell. Here's pinocytosis. So you see molecules, a vesicle forms around them and brings them into the cell. This is cell drinking or intestinal sipping. Exocytosis, on the other hand, is when a vesicle fuses with the membrane and dumps its contents outside of the cell. We know a lot about this because of our discussions of the cell and how it works, and we know that the Golgi body will take proteins, hormones, enzymes that are made elsewhere in the cell, and it will send them off towards the membrane in a vesicle, and the vesicle will fuse with the membrane, and the contents will leave the cell. So make sure you come to class with all your hot questions about facilitative and active transport.